Good morning. This is Dr.R.Kamalambigeswari, Associate Professor from Department of Industrial Biotechnology. Today, I'm going to discuss about the topic non-coding DNA. This non-coding DNA describes the component of an organism's DNA sequence that do not encode for protein sequences. That means these are the DNA sequences that doesn't have the ability to code for a functional protein. And in eukaryotes, a large proportion of an organism's total DNA is of non-coding DNA. And if we compare the amount of non-coding DNA among the eukaryotic species, of course it varies greatly, which depends upon the size of the genome of an organism. And the amount of non-coding DNA is always high when compared with the coding DNA as far as eukaryotes are concerned. And much of this DNA doesn't have known biological function, and hence it was referred to as junk DNA. And there are different types of non-coding DNA. Uh, the various types of non-coding DNA are non-coding functional RNA, cis and trans regulatory elements, introns, pseudogenes, repeat sequences, transposons, and telomeres. And let us discuss one by one in it, right? First, coming to non-coding functional RNA, the RNA molecules which are not translated into proteins are said to be the non-coding functional RNA. That means the DNA sequences will be transcribed to RNA, but the RNA that has been transcribed won't have the ability to get translated into proteins. And they will remain as RNA in the genome and they execute their function in the cellular environment. Example are ribosomal RNA, transfer RNA, microRNA, etc. The next type of non-coding DNA is cis and trans regulatory elements. The cis regulatory elements are those sequences that are present very near to the gene and can control the expression of a gene. Coming to trans regulatory elements, these trans regulatory elements are present far away from the active gene, but in spite of their presence, though it is present far away from the gene, it do have the ability to control the expression of a gene. They are located within the 5' or 3' untranslated regions in the genome. Next, coming to introns. These introns are said to be intervening sequences. They are the non-coding segments of a gene and they are transcribed in the precursor mRNA. And from the precursor mRNA, they will be removed through a process called RNA splicing, where the removed introns uh, will uh, get get detached from or will get itself removed from the mRNA and the exons join together to code for a functional protein. And next is coming to pseudogenes. Pseudogenes, as the name implies, they are said to be the false genes. They do have some structural similarity with the active genes, but they can't code for a functional protein. They may lose the ability to code for a functional protein due to various reasons like mutations and all. And they are called as genomic fossils. That means they remain in the genome as fossil. They are inherited generation after generation, but they don't have the ability to code for a functional protein. Next type of non-coding DNA is repeat sequences. The repeat sequences are those sequences that are repeated in the genome in many copies. These repetitive sequences are of two types, like highly repetitive sequences and moderately repetitive sequences. Both these highly repetitive sequences and the moderately repetitive sequences are distributed at irregular intervals in the genome, or else they may be clustered together also. If they are distributed at irregular intervals, then they are said to be interspaced repeats. And if they are clustered together, then they are called as clustered repeats. Next, coming to transposons, these are said to be jumping genes. They do have the ability to detach from one part of the genome and can go and attach itself in other part of the genome. And there are two different types of transposons, like class 1 transposons and class 2 transposable elements. And coming to class 1 transposable elements, they are said to be retrotransposons. Mm, they are of two types. One is said to be lines, the other one is said to be SINES. Line stands for long interspace nuclear elements, and SINES stands for short interspace nuclear elements. Both of these transposons belong to the class of retrotransposons. That means they use RNA as an intermediate for transposition. They account for large proportion of genomic sequences in many of the species. Next type of non-coding DNA is telomere. Telomere, of course, telomere are present at the ends of the chromosome, and they are highly comprised of sequences of repetitive DNA. The sequences that are present in the telomere is highly repeating in nature, and these telomeres are, of course, present at the end of the chromosome, and they are helpful for the integrity of the chromosome. Next, coming to the functions of non-coding DNA, they are highly conserved under evolutionary pressure, and some sequences are highly essential for maintaining the chromosome structure, as we have seen about telomere. The telomere, of course, it is it is the main part of the chromosome which is necessary for maintaining the integrity and structure of the chromosome. Likewise, the centromeric region of the chromosome is also comprised of non-coding DNA. So these sequences are highly essential for maintaining the chromosome structure. And some of the non-coding DNA may also serve as the site for binding of different transcription factors, which is going to uh, act on the expression of the gene. And some non-coding DNA will also serve as an operator for the activation of several genes, and they also serve as enhancers as well as silencers. And next is we will discuss about the DNA replication in eukaryotes. The DNA replication in eukaryotes can be studied in three different steps like initiation, elongation, and termination. And DNA replication is a process where the DNA is going to make a replica of itself. It is going to make an exact copy of itself. So, coming to the stages in DNA replication, um, the first stage is said to be the initiation of DNA replication, where the origin of replication is going to be is going to be formed at the site, and the replication is going to get initiated. And for the initiation of replication, as far as eukaryotes is concerned, the pre-replication complex formation is essential, which occurs in the G1 phase of the cell cycle. And the activation of this pre-replication complex that has been formed in G1 phase will be carried out in S phase. And for the activation of this pre-replication complex, two different enzymes are highly important. They are CDK and GDK enzymes. Coming to the cell cycle, as far as eukaryotic cell is concerned, there are four different phases such as G1, S, G2, and M. And in case of G1 phase, in the cell cycle, there are low level of CDK activity, which means the cyclin-dependent kinase activity. And while the CDK level is low in the cell, especially at the G1 phase of the cell cycle, the formation of new pre-RC complex is enhanced. Whereas in the remaining phases of cell cycle, there are high level of CDK activity in the cell, which won't allow the formation of pre-replication complex. So while the CDK level is high in the cell, then of course new pre-RC complex cannot be formed. And while the cell is going for transition from G1 phase of the cell cycle to S phase of the cell cycle, the S phase specific cyclin dependent protein kinase and DBF4 kinase, which stands for DDK, will transform this pre-RC complex that has been formed into a active replication fork. Thereby, the replication will be initiated. And this is just a representation of the CDK activity. Uh, while the CDK activity is low in the cell, then of course it enhances the formation of pre-replication complex, and the formed pre-replication complex will not be activated while the CDK activity is low in the cell. Next, when the CDK activity is high in the cell, then of course the new pre-RC complex cannot be formed, but the formed pre-RC complex will be activated, and hence the activation of pre-RC complex occurs in the S phase of the cell cycle, whereas the formation of pre-RC complex uh, will be will occur in the G1 phase of the cell cycle. Coming to the steps in the formation of the pre-replication complex, the first and foremost thing is that the ORC has to bind with the origin of replication sequence. So the recognition of the replicator by the eukaryotic initiator ORC occurs, and once the ORC is bound, then of course it recruits two different enzymes like CDC6 and CDT1. CDC6 stands for cell division cycle 6 protein, and CDT1 stands for chromatin licensing and DNA replication factor 1 protein. ORC and the loading proteins recruit eukaryotic replication protein, the helicase, the complex is said to be MCM27 complex. The association of all this protein occurs during the G1 phase and are activated to initiate replication after the cell transits from G1 to S phase. The activation of this protein has been related to its phosphorylation. Once the phosphorylation of this protein has occurred, then of course it results in the assembly of addition of the replication proteins at the origin sequences, and of course the initiation of replication at S phase occurs. And these new proteins include the three eukaryotic DNA polymerases. The DNA polymerases assemble at the site, and thereby the DNA polymerase alpha, which enhances or which has the ability to go for primase activity, will synthesize an RNA primer and just briefly extends the RNA primer. And hence, the initiation of replication has been commenced. And these are all the main events that occurs in the initiation of replication. First one is the recognition of origin. Next one is said to be DNA melting, where the double-stranded DNA is going to be melted into two single-stranded DNA. And the stabilization of single strand occurs, whereby the SSB proteins bind to the two single-stranded DNA that has been formed and stabilizes the DNA in the single-stranded form. And the assembly of chromosomes, that means various complexes that is necessary for the various proteins that are necessary for the replication, will assemble at the replication fork and enhances the replication. And the synthesis of two daughter strands has been initiated. So this is the eukaryotic replication fork that has been formed. The progress of the replication fork in eukaryotes is maintained by the helicase activity. That means as the DNA unwind each other, then the replication fork moves forward, and the separated polynucleotides are prevented from reattaching by SSB proteins, with the binding of single-stranded DNA binding protein. And the main single-stranded binding protein in eukaryotes is RPA, which stands for replication protein A. So if you see the elongation of the DNA replication, the leading strand and the lagging strand undergoing the process of synthesis. So the strand that runs from 3' to 5' direction is said to be the leading strand, and the strand that runs in 5' to 3' direction is said to be the lagging strand. In leading strand, the DNA synthesis is continuous, and the RNA primer is synthesized only once, and the leading strand synthesis is continuous by the helicase activity as well as by the DNA polymerase enzyme. And if you look into the lagging strand, of course, the DNA synthesis is not continuous here, and the DNA fragments has been synthesized as short fragments under the name of Okazaki fragments. These Okazaki fragments will be joined by the enzyme DNA ligase, and then it will be seen so that the lagging strand will be made continuous. And the RNA primer in the lagging strand as well as in the leading strand will be removed by two different mechanisms that is illustrated, say the RNase H model and the flag model. Coming to the RNase H model, the RNase H enzyme removes the RNA primer up till the last nucleotide, and the FEN1, that is the endonuclease enzyme, removes the last ribonucleotide plus some part of the DNA, and the DNA polymerase will will polymerize the removed nucleotides of the outer genome, and the DNA ligase seals the nick between the two DNA fragments. And coming to the flap model, the DNA polymerase delta and the helicase enzyme pushes aside the primer, and the FEN1 cuts at the branch point, and thereby the RNA primer along with some nucleotides will be removed, and the DNA polymerizes the cut removed nucleotide, and the DNA ligase enzyme makes the so seals the nick between the DNA fragments. Coming to the termination of DNA replication, the removal of the terminal primer at the end of the landing strand synthesis leaves a small gap that cannot be filled in if left unfixed, and this gap leads to the loss of terminal sequences. And this may lead to the end replication problem. That means the chromosomal ends may not be replicated sometimes. And this problem has been overcome by the enzyme telomerases. This telomerase, of course, it extends the 3' end of the parental chromosome beyond the 5' end of the daughter strand. It is a ribonuclear protein, and the termination of DNA replication is enhanced by two different factors, by the association of these two different factors, TRF1 and TRF2, which binds to the single-stranded DNA and plays a vital role in the stabilization of telomere and the regulation of telomere. And next we'll discuss about the transcription. Coming to the transcription, this is a process where the synthesis of RNA occurs from DNA molecule in the presence of the enzyme RNA polymerase. In other words, the process of formation of a messenger RNA molecule using a DNA molecule as a template is referred to as transcription. Coming to the synthesis of RNA, the RNA synthesis has been mediated by the RNA molecule. The RNA is processed into the synthesis of RNA from the DNA template by the action of with the help of the enzyme RNA polymerase, and the RNA is processed into the messenger RNA, the mRNA, which is then used for the synthesis of protein. And the RNA that has been synthesized is called as mRNA because this RNA carries the genetic message from the DNA to the protein synthesizing machinery of the cell. And the main difference between the RNA and DNA sequence is the presence of uracil in RNA instead of thymine in DNA. That means if thymine is present in the DNA, then of course it's replaced by the uracil base in RNA. The DNA strand is going to be used in RNA synthesis is said to be the template strand because it provides the template for ordering the sequence of nucleotides in an RNA transcript. Whereas the DNA strand, which doesn't take part in DNA synthesis, which doesn't take part in RNA synthesis, is said to be the coding strand, since because its nucleotide will resemble exactly as the RNA transcript, except the uracil replaces the thymine. If we look into the enzymes that are involved in transcription, then of course the process of transcription is catalyzed by the specific enzyme called RNA polymerase. And if we look into the type of RNA polymerase in case of eukaryotes, it is of different types, say RNA polymerase 1, 2, and 3. And moreover, this RNA polymerase is a multi-subunit enzyme. And DNA sequence is enzymatically copied by RNA polymerase to produce a complementary nucleotide in case of RNA strand. So in the process of transcription, the genetic information that is coded in DNA is copied into a molecule of RNA. The genetic information is transcribed, and it results in the transfer of genetic information from DNA into RNA, and hence the process is said to be transcription. So all cellular RNA are synthesized from DNA templates through the process of transcription, and the DNA regions that can be transcribed into RNA are called as structural genes. And the template strand is the strand from which the RNA is actually transcribed. And it is also called as antisense strand, since because it possesses complementary sequence with the RNA that has been synthesized, RNA strand that has been synthesized. And the coding strand is also called as sense strand, since because it won't take part in the process of transcription, and if it the coding strand will not take part in the process of transcription, and hence it is said to be sense strand, since because it possesses the sequences are similar with the DNA sequences are similar with the RNA transcript that has been synthesized. And only the template strand is used for transcription, and the coding strand has not been used for transcription, will not be used also. And only a small portion of DNA has been transcribed in response to the development requirement, depending upon the physiological need as well as depending upon the environmental changes. And coming to the mechanism of transcription, the transcription occurs in three different steps: initiation, elongation, and termination. And for the synthesis of RNA from DNA, the main enzyme which plays an active role is RNA polymerase. Because of this RNA polymerase, the DNA, as well as because of this RNA polymerase, as well as with the help of various transcription factors as well as proteins, the DNA can be transcribed to RNA. So this is the enzyme that is going to catalyze the synthesis of RNA, and it doesn't need a primer, rather than it can initiate the transcription de novo. The transcription can be initiated de novo, and it performs the same reaction in all the cells, from bacteria to humans. So this RNA polymerase is necessary for transcription of RNA from DNA in eukaryotic as well as in prokaryotic cells. And coming to prokaryotic cell, especially the bacteria, has only a single RNA polymerase enzyme. But in case of eukaryotes, as I discussed earlier, we have three different types of RNA polymerases, like RNA polymerase 1, 2, and 3. So among these three enzymes, RNA polymerase 2 is the most studied, and of course it's responsible for transcribing all protein coding genes. And polymerase 1 and polymerase 3 are responsible for transcribing specialized RNA coding genes. And the shape of this RNA polymerase resembles a craft group, and it is a multi-subunit enzyme. And coming to the pre-initiation complex formation for the synthesis of RNA, especially for the transcription, the GTFs helps polymerase to bind to the promoter and melt DNA. GTFs are said to be the general transcription factors, and they will help in the RNA polymerase to bind with the exact region called promoter, and and to melt the double-stranded DNA into single-stranded form. And the complete set of GTFs and the polymerase will bind together at the promoter region, and they pause for initiation, which is said to be the pre-initiation complex. It means the assembly of various proteins and transcription factors at the site of promoter for the initiation is said to be the pre-initiation complex. And many RNA polymerase 2 enzyme promoters contains the Tata elements, which is often referred to as a Tata box, where pre-initiation complex formation begins. So this Tata box or Tata elements will be enriched in adenine and thymine sequences. The sequences will be TA, TA, TA. The sequence will be repeating. The Tata elements recognized by GTFs called TF2D. So TF2D is the first and foremost transcription factor that recognizes the Tata sequences in the promoter and bind to the Tata box. The components of this TF2D that binds to the Tata DNA sequences is called TBP, stands for Tata binding protein. So the other subunit of this TF2D enzyme is TAF, which control the DNA binding activity of TBP. TAF stands for TBP associated factor. The resulting TBP DNA complex provide a platform for the attachment of other transcription factors as well as It enhances the attachment of the enzyme RNA polymerase. The factors TF2A and TF2B bind to this complex, that means to TF2D, where it has been bounded with the TATA sequence. And after that, the TF2F is the other next transcription factor that is going to get bounded with the TF2D, TF2A, and TF2B, along with the enzyme polymerase, RNA polymerase. At last, the two transcription factors called TF2E and TF2H bind to the upstream of the RNA polymerase 2 enzyme, which results in the formation of pre-initiation complex. So the formation of this complex containing all these components is followed by promoter melting. That means the double-stranded DNA is going to get unwounded to a single-stranded form. So this promoter melting in eukaryotes requires the hydrolysis of ATP, and of course it is the energy consuming process, and of course it is going to be mediated by the enzyme TF2H. So this TF2H is responsible for the helicase activity. It is going to melt the double-stranded DNA by hydrolyzing the bonds that is existing between the double-stranded DNA. So the large subunit of the polymerase 2 enzyme has a C-terminal domain which extends like a tail. So this C-terminal domain contains a series of repeats of heptapeptide sequences like the tyrosine, serine, proline, threonine, serine, proline, and serine. So this is the heptapeptide sequence that is present in the C-terminal domain, which is necessary for the functioning of the RNA polymerase enzyme. So the association of many different transcription factors is represented here. So the TF2D, in TF2D we have TBP and TAF. So the TBP is binding with the Tata box, which is followed by the binding of TF2B, TF2A and TF2B. So then TF2F comes and assembles along with the RNA polymerase 2, and so this is the RNA polymerase 2 enzyme, and the TF2F is responsible for bringing this RNA polymerase 2 enzyme for initiation of pre-replication complex, and the binding of this RNA polymerase enzyme results in the binding of the two more transcription factors like TF2E and TF2H. So all these transcription factors and the enzyme assemble at the promoter region of the gene, whereby the promoter melting occurs, which indicates the initiation of transcription.